Hello, my name is Marissa. I'm from Zimbabwe. I'm on Team S with my colleague Ahmed from Egypt. We are debating the topic, should African universities prioritize bioinformatics training over wet lab genomics due to cost and scalability? We will argue in the affirmative. I'll start with an introduction for context. Advances in genetics and genomics have been transforming fields such as healthcare, agriculture, and disease surveillance worldwide. Universities across the continent are working to expand scientific training while managing constraints financially and infrastructurally, and simultaneously navigating competing national priorities. Genomics education in Africa must be shaped by the reality of our contexts, our needs, our challenges, and our opportunities, and universities are faced with difficult policy choices. This debate examines whether prioritizing bioinformatics training over wet lab genomics across the continent's diverse educational systems is practical, sustainable, and ethically and strategically necessary for the future of genomics capacity in Africa. Our stance in this debate is in favor of the topic. We argue that policy prioritizing bioinformatics training over wet lab genomics offers benefits over the alternative in terms of cost and scalability. We present 6 arguments in support of this policy. Our arguments will focus on the following 4 points: the responsibilities that educational institutions have to their students, to industry at large, and to scientific advancement; the ethical and regulatory considerations that factor favorably into this policy; the contextual considerations that are specific to African applications and landscapes; and finally, we will point out case studies that feature cost effectiveness and scalability benefits. Our first argument is that universities have an ethical responsibility to maximize effective and sustainable usage of shared funding, which is often scarce. A focus on bioinformatics training presents opportunities for financial sustainability. The upfront costs of the initial setting up of bioinformatics infrastructure is more straightforward to quantify and predict, whereas wet labs require expensive specialized equipment, which increases the risk of cost creep. The running costs of wet labs include ongoing acquisition of reagents and consumables, and each new experiment requires a complete set. The running and maintenance costs of bioinformatics infrastructure are much lower, as infrastructure is reusable and open source tools are available. For these reasons, it is more cost-effective to provide bioinformatics training to larger cohorts than it is to provide wet lab training. A 2025 article by Tiberi et al. States that bioinformatics centers have become crucial priorities in universities in response to large-scale biological datasets across health, agriculture, and environmental science, all of which are key sectors for Africa. The authors cite success stories of bioinformatics initiatives that have been established in Kenya, South Africa, Morocco, and Burkina Faso. Our second argument is on Africa's data sovereignty and governance. Building African capacity for bioinformatics allows Africa to retain control of the full genomics pipeline, including genomic data analysis and the benefits from it. Currently, African wet lab researchers collect African genomic samples, then get sidelined from finishing the stories they start. Sending genomic data outside the continent means the narratives of African genomics are shaped by non-Africans and told on our behalf, sometimes even told to us about ourselves. Secondly, outsourcing analysis of data from wet lab genomics means intellectual property resulting from analysis of African data is held outside Africa, and the continent loses the opportunity to capitalize on and enjoy the value from its own data. Thirdly, externalizing African genomic data means we have no knowledge of or even control over any subsequent usage of that data without the consent of African participants and African researchers. It opens up both our work and our samples to exploitation. In an article in 2025, Kadim et al. Highlighted that outsourcing genomic data analysis makes African access deferred to foreign regulation for any further interaction with our own data. The authors also outline the African Bioinformatics Center for Omics initiative, which is based in Senegal and is aiming to ensure sustainable scientific independence for Africa. Our next argument is on graduate employability and industry readiness. Training students in skills with limited local employment opportunities would raise ethical concerns. Universities have the responsibility to maximize the benefit that both graduates and industry reap from curricula. There are more settings in industry for bioinformatics infrastructure than there are for wet lab environments. As such, there is higher capacity for industry to absorb bioinformatics graduates compared to wet lab genomics graduates. Technological developments tend to advance more rapidly than most other fields, and thus emerging opportunities grow at a faster pace in bioinformatics than in wet lab genomics. With the growing number of graduates produced by universities year after year, industry needs to scale to accommodate the influx of entry-level professionals. The less specialized nature of bioinformatics infrastructure compared to wet labs means when there is an influx, dry labs can be increased in number more easily than wet labs. In a 2023 article, Adenike et al. Point out that life science curricula that do not integrate computational and data analysis bioinformatics skills result in graduates who have knowledge gaps and are not competitive on the labor market. Our fourth argument is on resilience to resource limitations. Bioinformatics setups are more resilient to resource constraints. It is more complex to optimize wet lab genomics sustainably while maintaining reliability of outputs and results. Wet lab genomics require a full set of consumables for each experiment and for each iteration of each experiment, which is not efficient when resource limitations arise. Conversely, resetting a bioinformatics environment uses reusable infrastructure, and optimizing for a reduction in resources is less likely to impact results. It is more logistically feasible to coordinate and share dry labs for multiple trainees and trainings than it is to accomplish the same for wet labs, which are more inflexible in size, location, and equippedness. Under-resourced wet labs carry ethical risks that are more consequential than under-resourced dry labs, including compromised samples, compromised data, compromised safety, and compromised working conditions for researchers. In 2025, a 2-week intensive wet lab and bioinformatics training was held in South Africa. In a report by Agboli et al., participants appreciated receiving training in optimization of some wet lab protocols to take less reagents in less time. However, it was also noted that due to equipment constraints, wet lab training was taken in groups and participants did not have the opportunity to practice protocols hands-on individually. This left participants feeling limited confidence that they could each independently replicate the optimized protocols or teach these protocols to their colleagues at their institutions after the training. This intensive training was held for only 36 participants. University cohorts are typically much larger than that, and therefore even this optimized wet lab training scenario would evidently not be scalable to a satisfactory university curriculum. Our fifth argument is on portability and continuity of work. Collaboration is an integral part of the work in both wet lab genomics and bioinformatics training and research. Bioinformatics environments can be replicated more reliably than wet lab conditions. This means training can be provided with uniformity across universities or across student cohorts at a university, and it makes reproducibility of bioinformatics work higher than wet lab genomics. Bioinformatics research can be extended by others more readily, as they can continue directly from where previous researchers left off with minimal repetition of existing work. Wet lab researchers may need to first replicate previous work in order to obtain the starting conditions of their own extension of the work. Coordination and harmonization of policies across jurisdictions is more attainable with bioinformatics than wet lab genomics training, as the regional agreements for sharing analysis are less complex than those for sharing samples. In a 2025 article, Lukele et al. Share case studies of bioinformatics initiatives in Africa, including H3A Bionet and the Eastern Africa Network for Bioinformatics Training. Both of these initiatives have provided and supported bioinformatics training within universities, research collaboration across institutions, access to computational resources, standardized bioinformatics university curricula, and infrastructure development. Our sixth and final argument is on adaptability to evolving science. Universities need to prioritize investment in future-proof facilities and teaching of future-proof skills. For instance, our understanding of the genome itself has evolved before and may yet evolve further. Bioinformatics training is more adaptable when scientific understanding shifts. Wet lab experiments may need to be redesigned or repeated, which is costly and time-consuming, whereas bioinformatics supports reinterpretation without re-experimentation. Bioinformatics infrastructure is transferable across changing scientific priorities, and shifts in analysis direction can often be executed with the same underlying infrastructure implementations. The equipment and environment of wet labs are more likely to be closely tailored and aligned to experiments and their objectives. Bioinformatics skills in data interpretation and algorithmic and computational thinking continue to retain relevance and value even when foundational genomics methods evolve. This is not a flexibility that wet lab skills share. Policymakers can't predict which biological and other scientific theories will continue to prevail as understanding evolves. Policies that promote flexible analytical capacity will be more resilient over time. Deshpande et al. Indicate in a 2024 article that computational research is efficient, allowing for hypotheses to be formulated from insights derived from data interpretation. Wet lab research is hypothesis-led. It uses specialized equipment and is thus less flexible. The team opposing the topic has put forward 5 arguments. We provide our rebuttals for each one in turn. Their first claim is, bioinformatics depends on wet lab genomics. Our response is that prioritizing bioinformatics does not eliminate wet labs. It addresses the current bottleneck where data exists, but local analytical and computational capacity is insufficient, as mentioned by the previously cited Kadin et al. Their second claim is, wet lab genomics ensures scientific sovereignty. Our response is that scientific sovereignty is in what happens to our samples after collection. Loss of ownership in the past occurred because local wet labs existed but outsourced the analysis, and Ochola makes this point in a 2025 article. The third claim is, local relevance requires local data. In response, we note that it is true that Africa has high genetic diversity. However, neglecting local bioinformatics capacity means when we collect African samples, we begin to look externally for analysis to have who we are translated to us by others outside the continent. The fourth claim by the opposing team is integrated training builds better scientists. Our stance is for prioritization of bioinformatics rather than exclusion of wet labs. Integrated training is ideal. However, with resource constraints and a specific focus on scalability, prioritizing bioinformatics corrects the current imbalance where wet lab skills exist without analytical depth. And again, we cite Kadim et al. Initially prioritizing bioinformatics and then moving on to wet lab expansion as a second step ultimately supports stronger integration. The final opposing claim is, wet lab genomics drives innovation and jobs. Our response is that while employment opportunities exist for both bioinformatics and wet lab graduates, there is a mismatch between qualification and skills obtained in wet lab genomics versus labor market requirements, as highlighted by Adeneke et al. In the case of curricula in Nigeria. In closing, African universities operate in environments of constrained resources but immense potential. With limited resources, prioritization is inevitable rather than optional. The question is whether African universities will prioritize in ways that are forward-looking and maximize the scope of impact in terms of affordability, the number and diversity of students and graduates with genomic education access, the number and diversity of institutions that can participate in genomic education and research, the longevity, relevance, and value of the skills that are taught, adaptability of infrastructure and curricula, the extent of African scientific self-determination, and the number of African communities that ultimately benefit from African genomic science. Policies prioritizing bioinformatics training will empower these stakeholders, both in the present and for future possibilities, without having to wait for expensive wet lab infrastructure to mature. Through bioinformatics, African universities can move from limited participation to leadership in the genomics era. The floor is now open for any questions on our presentation. Thank you.